they come in. Hi, everybody. My name is Scotty Sims. I am the Executive Director of the KCNQ2 Cure Alliance, and we are glad to have everybody here today for this webinar. Just a few quick housekeeping rules. We are going to have a presentation first, and we ask that everybody keep your microphones muted, please. And afterwards, then there will be a Q&A session. We're going to take questions. We'll call on names so people aren't speaking over each other. So we just ask that you either use the react button at the bottom of your Zoom where you can raise your hand, or you can physically just raise your hand and we will call on you and then you can ask your questions. All right, I am going to call out Brooke Babineau, who is our science director, and we're super excited to have her on our team, and she's going to be telling you a little about the webinar and introduce our speakers today. Thank you.
Great. Thanks, Scotty. And so great to see so many of you here. As you've probably noticed through all our social media pushes, we're really trying to take KCNQ2 research to the next level. And that requires you all and participation from you who live this every day. So thank you so much for being here. I'll keep my comments relatively brief. But I want to introduce Dennis Lahl, who's an associate professor at UT Houston, as well as the director of Center for Neurogenetics. And his team is here as well to explain some of their really exciting work in creating a portal that is cross neurodevelopmental disorders, how both interview and survey can feed into that, as well as some other really exciting studies. And I just want to also call out, instead of listing all of Dennis's and his team's many accomplishments, I think what's most important for us to know is that he has really partnered with multiple different patient advocacy groups and is really working towards standardizing data in a way that we can make really big and broad conclusions. So I think being partnered with his team is a really important step forward for us. So I'm grateful to have you all here. Dennis, I'll turn it over to you.
Well, thank you so much. Yeah, and super excited to have everyone here. And I will directly jump in and and try to keep it brief, which is hard for me. I talk a lot, but I would try to do my best, and then we can talk a little bit all together. Then in a second about questions you may have. Okay, can you guys see what I show? I see a couple of people nodding. Good. Okay, so we can just directly jump in. So we are building a so-called KCNQ2 portal, and this is on purpose say we are building, we're not saying we want to, but we are building. So that is supported by the foundation, and we are very grateful for this because we really like to do these things, and we have like the last weeks were really amazing, and last months actually, and all the support we got, and the goal over the next weeks, and of course longer term, but as particularly the next weeks, is to really build the best portal possible that we can present even at the summit in Philadelphia. And it's really great that you have many of you already joining us today. Okay, so what is a gene portal? This seems to be very obvious, but at the same time also very important question. So just to keep it simple, you may have noticed that many of your providers or physicians you work with, they're not very informed about what's going on in KCNQ2 research. You feel this sometimes when you have questions and the providers don't have answers. And at the same time, also a lot of the clinicians sometimes struggle a little bit understanding molecular data or clinical data, or just in general, trying to have challenges keeping up with what is happening in research. And the goal of a portal is to have what I wrote here, have everything in one place. So if you want to learn if there's new knowledge or what is the best knowledge at that point in time, let's say today, you want to only go to one place, and you don't want to have to read hundreds of research papers. And you also, if you want to have a question and you want to learn about the disease or about the data, about mutations, what they do, you want to be able to interactively explore this. And this is important for families just to learn about the disease, for example, newly diagnosed, or if you want to know how do older individuals look like, or what do specific mutations do in terms of function. But it's also important for clinicians, because most clinicians are not trained in KCNQ2, and they need to really have a resource where they can go on a regular basis. This is the same also for drug developers and clinic, academic or non-academic. And so we're living now in an age where the medical knowledge is doubling every 8 years, and it's really important to access information fast and effectively. So in general, just having one place where you can learn about what's going on and about new knowledge and KCNQ2. Okay, so what Brooke already mentioned is we have built already several portals, and there's more in development at the moment. And each of them represents always the largest data source for any of these disorders. And the numbers are already, for some of the portals, are reaching already a couple thousand, whereas for others, they're still growing. And my gut feeling is that for KCNQ2, we will very fast be one of the larger ones, just based on as a researcher, what I have observed the last years, and also this very amazing engagement we got already from people who registered the last week. So, and I come to more about this. So here's now an example how the experience can look like about in a specific portal. So here's the GRIN portal. It's an older version, to be honest. But here, one of the features which I want to highlight particularly is the variant interpretation piece. So for example, instead of KCNQ2, we have, the GRIN2B gene. Because I show you this because it's one of our most advanced portals. And here you can enter a variant or mutation, depends on how you want to call it. And then you get a lot of information. And this is information which is really important in terms of understanding, is the mutation or variant pathogenic? And because this can change sometimes, it's not that the mutation changes, but our interpretation changes. And also what the mutation does in terms of functional consequences. And so having a resource publicly available, which has always the largest patient data and largest functional data available publicly, helps a lot of the genetic counselors, geneticists, and the more genetics heavy or savvy neurologists to give good diagnoses. And also gives you also a way to confirm that the interpretation which was done by the diagnostic lab makes sense. And if not, you may ask then other people what's going on. So because sometimes, test interpretation is sometimes still a challenge. And here you can see, for example, this variant here in GRIN2B has been functionally tested. And there's a lot of other tools which you can explore here, where you can learn more about the variant. But I don't want to go into too much detail here, but the most important thing, it helps with diagnostics, because nothing is more important in diagnostics is knowing if the mutation has been seen in other patients. And if you want to even think about prognosis, so if you want to ask the question, how do people who have the same mutation or variant typically look like in different ages, when they're young or when they're teenagers? Also, having patient data from nothing is more important than that. And so that's why we need to collect data and make this accessible, that one can learn about these individual variants. Because the more we collect, the more we see that that the variants are not so unique than we originally thought. For many of these variants, we see now many patients over time, but it's a big effort to get all the data together. Okay, so I will move on here, and we can talk more about the details later. Okay, so but there's also other features. So you can see here that we have here on the top left that we show for the SCN portal is a different one. What's the typical clinical spectrum? What types of when do typically, for example, here the seizures start? Is there a difference by type of mutation? Then for researchers, we have these interfaces where they can explore the data and they can even do research with these websites. And this sounds trivial, but many of the molecular researchers, they are not so good at combining it with clinical data. And the clinical people, they typically have challenges making sense of these molecular data sets, such electrophysiology, and others have problems with matching all the information together on the protein, so basically the channel. And with these interfaces, we provide this all for the community, that they can just explore and click, and you can even do this, right? So there's no one more qualified than a very motivated parent then explore information about the child's mutation. Of course, it requires certain training, but at least it is in public space. Everyone can access it. And this helps then also companies who may consider developing a new drug program to just explore all the data there is, and you don't need to ask scientists for permission or so. So overall, the idea is that it speeds up the progress in accessing the data. So here's an example of users from our portals. So the other gene disorders where the portals are already out. And you can see that they have been that they are widely used. So every red dot is like when people use them. So however, we see that we need to translate the portal in more languages, that we have more people accessing them. Okay, so the biggest factor is, and this is also, I think, probably a big difference compared to existing resources for KCNQ2, is the whole idea is to make everything directly accessible from the get-go, so that the data is not behind some kind of user accounts or so. Everything is de-identified and publicly available, so that scientists, but also citizen scientists like you or parents or any other person interested, can directly explore the data and just by clicking get statistics and all kinds of information out. So, and overall, the goal of these portals is really to be a resource where at any point in time you have the biggest patient cohort data, the biggest functional data, all curated, accessible, and at any point in time, so that you don't need to look at publications or other resources. And so it needs to be continuously updated. But if we achieve this, then this should lead to better diagnosis, better care decisions. And this is particularly important for the current age of precision therapies, which are upcoming, and and also facilitate research. Because for many of the new therapies, you need to know, is it gain of function, is it loss of function? And for this, you need to have decision support, and we can see this already in the other tools. But having all the data combined together, the genetic data, the clinical data, and also the molecular data, we can actually figure out new research. And this happened in other projects where we developed the portal, and suddenly we noticed some interesting patterns, and that led to research which we originally didn't plan. And this typically happens actually for every portal. Okay, so what type of data can go into the KCNQ2 portal? And basically any kind of data which is already collected. And so on the one hand, for example, the Ricky database with Dr. Cooper, we are actively working on trying to get the data transferred. It's a little bit challenging, but we are really working on this. So then also natural history data from, for example, Dr. Puduri, then data from electronic health record sharing initiatives, for example, the Citizen Initiative, and of course, clinical and research data from the literature. But at the end, any form of data where the family consented for research, that it can be shared, de-identified, typically, there can be included in such a portal. And today, I also want to talk a little bit more about data which we want to generate with you over the next weeks. So, so we have also a couple initiatives going on. Two of them are remote, which is, for example, the parent interview, which is a remote, and the REDCap survey is also remote. And then we have the clinical neuro exam and wearable study, which is on site at the summit in Philadelphia. So just to very briefly scratch on this. So the parent interview is you can participate simply by scanning the QR code, and then you will go to a website where you can sign up, and then you have the opportunity to talk to Mariana, who is also on the call. Maybe Mariana, you can briefly say hello.
Hi.
Perfect.
Yeah,
And
so if you've signed up, you'll be getting an email from me soon to get that scheduled for the interview he's talking about.
perfect. Thank you so, Mariana. And I think in the Q&A session, we will talk more about this. Probably there's more questions. And so the parent interview, just give you a little more detail. It takes about 30 to 60 minutes, depends on how chatty you are and how complex also the medical history is of your loved one with KCNQ2. So it's unstructured, which means or it's semi-structured, but it's almost unstructured. So what it means, we will ask you, hey, can you tell us about your experience with your child or your partner, who depends, with KCNQ2 related disorders at birth. And then you can just tell if there was anything you feel which you feel was related to the disease. And you just share your expectation or your observation. And for example, for someone where, for example, having a child who had neonatal seizures, you probably will say a lot, right? So what happened during that time, potentially in the NICU, and you will talk about this. But for someone where maybe there were no neonatal seizures, you may say, yeah, there was nothing, but I noticed this. And you're free to share whatever you feel is you want to share, because for some of the things, they only emerge later on, if we hear this from many, many families, that this is actually a part of the disease. So for example, for other disorders, we are noticing now that the children had problems latching on for breastfeeding already in week 2 and week 3, and the parents needed to come back to the hospital to get feeding support, and then it resolved. And since not every family had this, it was not noticed by the doctors, and it was not noticed by the family that this is part of the disease. But we have heard the same story many, many times now, and we feel now, oh, okay, this is actually a particular part of the disease. And this comes only true if we talk to many, many families. Just as was one example. So, and then in this case, Mariana would ask you then later on, what's happening between the first week of life and month 3, and then what happened between month 3 and the first year, and so on. The interview goes until the age of your child today, and you can just share what you want. So, and then we analyze the data later, and this gives us then insights. How do these insights, for example, look like? So here's now a poster which we presented at the SCN2A conference like 2 weeks ago in Colorado. And this is just data based on 12 parent interviews on their children. And we see, for example, what I just mentioned, this difficult to latch in the newborn period, also already sleep problems, and then also interestingly missing, or just tiny bit missing the milestones, which didn't came across because this is hard to capture in surveys. And also like a lot of contextual information. So I cannot go much into detail. The scientists always want to talk about everything forever, but just happy to answer questions later on. So, but these interviews already in small numbers are incredibly useful. So I'm a scientist, for example, studying SCN2A since 15 years, and there's hundreds of papers. But what these 12 parent interviews showed us on several levels is not in the literature. It's very powerful. Really encourage you to participate. Okay, so the next thing is the REDCap survey. So the REDCap survey is asking up to 180 questions. I know it's a lot, it's a big commitment. Up to 180 questions about when did a symptom start. For example, if your child has seizures, we will ask, when did the first seizure happen? What type of seizure was it? But if your child doesn't have seizures, then you probably only have to so you don't have to answer all these detailed questions on seizures. So long story short, if you have a child which has a lot of medical needs, probably the REDCap survey will take you also quite a while. So probably an hour or so, depending if you have all information in the back of your head. But if your child is relatively mild, it's relatively fast. But the good thing is these survey is really structured data. This helps us really fast to analyze it, and we want to present data already at the summit. So this is really important. So we want in the next weeks, we really, every time someone fills out this survey, we directly analyze it so that we can present the data. And so how does it look like? So we have done this already for around about 2,000 patients in collaboration with the Simons Foundation. And when we, for example, have the data can look like this. Imagine instead of here now the 16p11 deletion, here would be KCNQ2. And what would you get out of this? We learn now what fraction of people with, for example, KCNQ2 have certain clinical needs. For example, I pause here now, just so you can see it briefly. Okay. So among respiratory problems, which we may ask, depends if you what you click. We see, for example, that this amount of people have other respiratory problems. It's a bad example. Let's go here. Okay, so what fraction of people have seizures? And so these kind of statistics, each bar showing percent, we will have for KCNQ2 at the summit. These statistics get better the more parents we have who fill this out, because with more people, we can actually split by type of mutation. And we can also split by, is it a boy or is it a girl? And there might be differences which have not yet been understood, but they they emerge when the moment we have data. And so, and here's now, and where I want to go a little bit more in more detail later on, is we can also calculate these type of curves here. So in this curve, because we ask always, when did it start? And here's now an example for SSC6A1, but keep this in the back of that, could be KCNQ2 in 3 weeks. The moment we have the data, we can do these kind of plots. So you can see now, they're complicated plots, but I will just briefly try to explain one. Where you can see, let's see where I stop hovering here. Okay, so, okay, it didn't work. Let's see. Okay, so here's a good example. So we might see something for KCNQ2, which is interesting, like here in this example for SSC6A1. So the pink graph means this is SSC6A1, and the gray one means any of the other 2,000 children with neurodevelopmental disorders. And we see among people who have absence seizures, that in SSC6A1, first of all, everyone has it. So this is, but not everyone gets it directly at birth. So for example, 50% of people with SSC6A1 have absence seizures by the age of 2 years and 3 months. But there's also others who develop it later. This is the later phase here. So basically by the age of 10, everyone has developed it, but not by age 2. By age 2, it's only 50%. And just imagine this level of information would be available when you have gotten the first time to a geneticist or your neurologist. So the moment we have this type of data, the clinicians, they can just go to the website, and the parent may ask, hey, my child has not developed this or that yet. What is my risk? Because I read online that this might be part of the disease. And at the moment, people just have to guess, or you have to hope that your doctor might know a paper, or they have at least seen 3 patients and they can just do some voodoo and remember it, whatever. But with these kind of online resources, clinicians can go to the website, or you can go to the website. You don't even need to directly go to the clinician, and then explore a little bit, and then you go to your doctor and discuss it with your doctor. This is, or you can just point to the doctor, can you explain this to me, what does this mean? So with this kind of information, you definitely get more data-informed information about when each symptom emerges and when the risk stops. For example, here on the right side, you see with my mouse that atonic seizures are across neurodevelopmental disorders are relatively rare, but in SSC6A1, it's more frequent. Like 40% of people have it. But what you see with this line here is basically people or children have developed it typically for SSC6A1 by the age of 5 years and not later anymore. So again, if you fill out this the survey, we can calculate these plots and learn about KCNQ2 related disorders. And we even can split, if we have many people participating, we can even split by different types of mutations. If they are in the middle of the channel or if they're more outside of the channel, what type, and other factors. And we can also look at, so we can look at many other factors. Okay, so the last thing what we will do is we will do clinical grade neurological exams at the summit, where we and do something like this. This is pictures from other conferences where we have participated. And so you always see people with smileys. These are children, and then members of our team who do neurological exams or record and also record these neurological exams with cameras that we can analyze the data later on in larger groups. And we also ask the families, in particular the children or the affected individuals, can also be adults, to wear these Empatica Embrace 2 watches, which also the astronauts from NASA at the moment wear in space. So these are these watches. They can measure the skin conductance, whatever that means. It means it's a proxy. People say it should measure if you're excited or not excited. So like parasympathicus and sympathicus. We're exploring this if that's actually true. Not so sure. Then it measures the heart rate, and for this it's actually FDA approved. So this is pretty good. And it can also measure the movement, so the accelerometer. And with this, we look at the moment in the 100 children, which we already, with neurodevelopmental disorders and epilepsy, which we already collected from these other conferences. We're looking at the moment if their movement is different compared to siblings. So we try always to get the sibling as well, or other children as a comparison group. And if they move less, if the sleep is different, because these watches are supposed to measure sleep, and other things. So then we compare this also to these clinical grade neurological exams. You can see, for example, here Sunny doing an exam with this child here where she's lying, and here he does like another exam with this girl, and here's more of this. So we hope that we can develop a way to measure the progress of the disease and the severity of the disease in the future, where we can just send you a watch and you wear it at home, so that you don't need to go to the doctor. And we have something objective to measure, because a lot of problems which we have at the moment with clinical trials for these new type of therapies is what to measure beyond seizures. How do we measure that someone is improving in their daily activities and their abilities? And for this, surveys are not so good. And we hope that the wearable could move into that direction. And so this is then very, basic research to move into that direction. Okay, so we do these neurological exams, which are really clinical grade, and then everyone is wearing also the watch, and people participate between the age of 2 and older, basically. That worked so far quite well. And then we perform these different type of movement scales. So these are clinical grade exam, which are well established by movement disorder specialists. And then we can get analysis like this here. For example, we can see here's controls, and you can see that here's no big bubbles, meaning these are the siblings which typically don't have movement disorders. And then we can see now for the children with these genetic disorders that they have a lot of challenges they face, and we can compare and describe them. And this has never been done, because typically when researchers describe movement disorders, then they extract it from their charts. And they don't do this in a standardized way where they look at many children in a very standardized setup that you can compare them. But since we have done this now many times, we can compare, for example, people with a KCNQ2 related disorder versus an SCN2A related disorder, and so on and so forth. And we're just expanding this continuously. Okay, so with this, I will close. I'm certain that there are hundreds of questions. The only point I want to make, as long as I have the mic alone, so it's super important, everything you contribute matters. I know it's participating in research and takes a lot of effort and time, and you already have you know, a lot of things going on in life. And when we collect data, we can and will directly analyze it, and which will make the difference. And yeah, really appreciate if we have data to put into the portals or just analyze to try to, you know, identify new research knowledge. Thank you.
Thank you, Dennis, so much. That's wonderful. So we're super excited about this. This is our way as families to be able to move the needle forward. And as most of us remember, when our children were diagnosed, there was such a long period of time waiting to find out details about our specific variants. And this is something that, as we can move research forward, it also helps families to prevent, hopefully, those prolonged periods of not knowing. All right, does anybody have any questions regarding the presentation, anything about data, how to apply, anything at all? Brian?
Here, turn on my video. I had, of course, I'm me. I had a list of them, but one of the things that I'd just love to see as this develops is that, you know, there was the parent survey probably 5-7 years ago, and as you ask questions about the percentage of patients who have attained certain developmental milestones, for instance, how many kids learn to walk, how many kids talk, how many can tie their shoes, go read books, etc. Because the very nature of genetic testing means that we have so many young patients in KCNQ2 world. When it's presented, not the data itself, but the presentation often skews, sorry, this is so long, it often skews that it shows, hey, not that many attain this, not that many have attained this. This percentage of parents say their kid doesn't do that, because the questions are being asked to people with 2-year-olds and 4-year-olds. Now that we've got some older kids in the data set, and now that we're getting a new data set, I'm really excited to see when this data is presented, of children who are over 5 years old, this percent are able to do this. Of patients who are over 10 years old, this percentage are able to attain this milestone. I'm hopeful that this new data set can gain some of that information, because it's that's one of the things that a new parent really wants to know is, what are the odds my kid's going to walk? What are the odds that my kid's going to go to school? Things like that. So yeah, that's one thing to keep in mind, that I'm really excited for a new data set to be able to ask all these questions again and be able to say, hey, these are, and when you present this data, it would be great to see those older kids are able to do this, and to kind of break that out by age rather than just asking a bunch of 2- and 3-year-olds parents, if that makes sense.
Yeah, it makes a lot of sense. So like, just direct follow-up. So please, everyone who's here, right? So we are developing these resources not for us, right? So we're developing this for you, and everyone is welcome to give feedback and design these portals jointly, and say, hey, this should be shown, and this should not be shown, and this should be better documented, because you know, we have no clue, right? So the more good feedback, like from Brian, the better. And at the summit, we can present the statistics you just suggested, and hopefully we have a prototype running so that you can also explore in parallel things yourself.
Scotty, do you want to continue to call on people, or?
Sorry, I was muted. Yes, Sky had her hand raised.
Thank you. Yes. So similar, I think, kind of vein as to what Brian was getting at, but what is going to be the process for the evolution of this data? For example, I'm a parent and my kid's 1 year old, and I want to go in and put in information, right, about my specific variant, but then maybe I never log in ever again. And so maybe my variant is more rare. And so one day someone comes along and they only have a little bit piece of my story. So the evolution of the data, like, is there kind of a thought given to what the evolution and the sustainment of how we continue to keep parents, like, coming back to this portal to keep providing data such that we know maybe things change, maybe things improve, that kind of scenario.
Yeah, so the most important thing, and this is what Mariana will mention to every parent who is, so first of all, to participate, you will have to meet Mariana. That's unfortunately the truth. And because we need to consent everyone so that everyone is really aware with data sharing and so on, and also for the interview in particular. But we will always ask or we will always try to almost insist if you could also generate a crit, which is like an identifier. So which is you generate basically a specific code, which is a marker for your child. And in research, more and more different research groups are using this in their surveys. So even if, let's say, we would close shop next week or so, the moment you fill out the information with the crit and you choose to participate in the future in a different project, our data could be used in the different project. So, so why do I mention this like this? As you saw, we are involved in many, many projects of developing portals. And as it turns out, this is the same like with every adapter ever built for an iPhone, for a USB stick or so. Everyone wants to build the one and only version of it. And then what happens is that you have more different adapters, more different surveys and so on. And so our goal is to aggregate everything there is at the moment and build out our survey, but also update the survey, reach out to families in a year from now or longer. Hey, would you fill this out again? But if, for example, there is a different source of information, we would also love to integrate that. And so in the future, for example, so having this identifier is super important. But I have to speak a little bit cautious because in the US system, there is no longevity in terms of resources for building in these kind of surveys for 10, 20 years. So we are actionable now. We have kindly received resources from the foundation, and we will aggregate everything there is and issue more, and are planning to issue more surveys and also more types of analysis, like the hopefully soon at home monitoring. But it's hard to predict the future. But the one thing what is super critical for every one of you, if you participate and people ask you for this so-called crit, it's incredibly important to generate it, because then this data we will know this is patient 500, and patient 500 can be identified in future studies, and you can compare the past and the future and learn from these resources, if it's our study or in a different one. Or even clinical records can be compared against this.
And I think to Dennis's point is, like, if you have a child with a common variant and maybe they have a different presentation than other children with that same variant, it's super important that you participate and have that crid, because what it does is then it connects your records and it has your child stand out individually. I mean, they're still de-identified. Their, identifiable information isn't shared, but they're still connected. And then if you participate in like Citizen Health for the medical records and you add your crid to that, and we're going to have something coming up soon on how to do that, then your child's medical records will connect with your responses on Dennis's survey. And it keeps your child's unique journey specific to them, not to the variant in general. And so it's also important for those who have a variant that's not common, or maybe the only person known with that variant. I think Martha's had her hand up.
My question goes for the watches. Are those only available for the summit, or is there a way that insurance can pay for it so they can get data for the ones that can't make the summit?
Yeah, so there was a little bit issues with the connection, but if I understand correctly, the
Did
watch.
you hear me?
Yeah, I think so. I think I got it. So you asked if the watches are only used at the summit, or can you also, for example, get them at home for insurances, right? I will just start talking, assuming
Yes.
that this was the question, because okay, perfect. So at the moment we are only doing the watches at the summit, because you can buy them as a private person, I think, but they are only made for research. So there is nothing on the watch besides the time, and to extract the data the watch is collecting, you would have to be able to program or code. So it's really a research tool. But that said, so we are trying to at the moment to analyze all data we have to make the case for NIH that this is valuable information, or pharma companies who want to move into clinical trials in the future, that we get resources either from pharma companies, for NIH or philanthropy, to start a really big study where we can monitor at home, and then we can just buy these watches and send them to everyone. But this requires a lot of coordination, because a watch alone is also not really useful. You also need to learn a little bit from a brief survey in parallel, or a diary or so, what happened during the time the person was wearing the watch, and some other factors. And so it becomes relatively costly in doing this type of research. And at the moment, the goal is mainly to describe in a small pilot study for every disease what we can learn from such a watch, and in parallel do one big analysis and use this information then to justify a multi-million-dollar grant. And this is needed because of the coordination, and the analysis time is quite a bit. And one single watch costs $3,000. It's like a little
Oh.
Rolex.
Thank you. Thank you for that.
We had a question in the chat asking about Ricky, and if participation in Ricky, if you weren't able to participate, will prevent you. This is completely separate from Ricky. What we're doing is we're actively working with Ed Cooper, trying to get the information from Ricky over into the KCN portal so that it is publicly available. So it's completely separate, but we are having those conversations on how we can bring that over. So again, I know when they did the initial consents for Ricky, they weren't really asking for a CRID, but that's something that we're looking to see if we can, for families that did participate and do have records, if there's a way we can do some reconsenting and get that information. But that's kind of down the road and separate from this. But no, and I think the big thing, and I've had several people just message me, like if you're looking for like level of commitment on this, is sign up and do your consent with Mariana, and get the questionnaire out. That gives us our real that kind of quick and dirty information that they can analyze really quick. And then, commit to what you can with the interviews. It'll take longer, as Dennis said, to pull that information out. That's more cumbersome, but the questionnaire is quick and dirty, and we can really, really start to generate our portal and get this information out to build that overall picture of our, of KCNQ2 and the phenotype.
Yeah, and just I mean, I feel very, so just you know, I know how precious time is, you know, particularly if you have to care for a family member with, you know, KCNQ2 or any neurodevelopmental disorder. I'm advisor on around about 15 parent organizations, so I have a little bit view on this. And I feel very strong and proud to say that our lab is publishing 15 to 30 publications a year. And we have built many portals in the last 3-4 years alone, and we are building more. So every effort you know, invest, we have a large team here from Mariana, Tobias, Sejong, and Emily, and many more which are not on this call, who will really proactively and fast work on this. So really, and you know, this is something which will directly translate. So every family who participates, shares information, will provide a resource for better in care, on better information and better knowledge about KCNQ2 for the next family.
And Dennis, I don't know if you can see the chat. There's a question.
Okay, I can read. So how do you ensure that there is statistical significance for the results generated by the data analysis? You mentioned that you presume that there will be a substantial amount of data for KCNQ2. However, especially initially, there may be only few data points for a particular variant. This could potentially reduce misleading results or sample size. But yeah. So, I mean, very valid, right? So the good thing is in the survey, we have data from 86 neurodevelopmental disorders from more than 2,000 people, children mainly, from other neurodevelopmental disorders. So if there is, for example, high rate of vision phenotype, let's say 10% of people with KCNQ2 have it, but among 2,000 people with the neurodevelopmental disorders, only 1% have it. Something like this becomes very fast significant in terms of statistics. But just to be very clear, and we at the beginning we would just show descriptive statistics. We show these bars and just say, hey, 10% of people with KCNQ2 who are younger than 6 years old have this. By the people with KCNQ2 from 6, or teenagers with KCNQ2, we observe that. And this is just an observational. It's there's no claim behind it. And it's, but I think, you know, but I know it's still informative, you know, just to see where things going. And the portal will be documented to the degree that people can then conclude themselves, you know, if this is, you know, valuable or not. But we will document, you know, limitations and strength of the data. And of course, if you want to know for a specific variant, what does this mutation or variant do particularly, this is something which takes a little bit longer because you need to go up to numbers that you have 10, 20 people with the same variant. And this is not so straightforward because this requires many people to participate. But over time, this is actually happening. And the good thing is we have also people like Tobias on the call, who is a bioinformatician and structural biologist. So he can characterize variants not only by the label, let's say amino acid position, blah, 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 but he can also look where is the mutation located on the ion channel and can find other mutations which have a different position but have the same function. For example, if you think about as in a car, if you have, you know, you have the brake left, the brakes on the left front tire are the same brakes on the right front tire, because we both times know that they are brakes and they work the same way, although they are at different positions. Does that make sense? And Tobias is in the position that he can say, hey, this position 400 is actually the same position as position 300. And so based on, for that reason, we can analyze them together and suddenly see patterns. So we can bring a little bit biology also in the interpretation of the clinical data. And this is actually our specialty. So we have done stuff like this a lot in the past, where we try to combine clinical data, with functional data, with molecular data, with protein structure data.
And there was a question just about the CRID. So just for clarification, the CRID literally is you going in and putting in, you know, your child's name and date of birth, and it creates this unique, it's a unique identifier. So it's a number that is specific to your child. So whenever you participate in research and they ask if you have a unique identifier, you add the CRID. And then what it does is, as all data is pulled together, depending on how it's done or who is doing it, your complete picture of your child is there. So the CRID itself is just a number. It is not a portal. But like if you participate in Citizen or a natural history study with Ampidori, or if you've been to summits in the past and you've given samples for our biobank at Combined Brain, your CRID connects all those together. So you have all your child's complete history connected to their variant. And so I hope that answers the question about the CRID. I just want to make sure it takes 2 minutes. It's just super fast. You just click on it, and it's international. It's accepted in the EU. It's a valid, unique identifier. Then Brian had asked about just like getting information and the financial kind of model for these various other organizations that do research. So, when we work really hard to make sure that when we're partnering as a foundation, that we're partnering with other people, that the foundation either has kind of owns the data so that it's not then taken in by an institution or somebody and then held under lock and key and cannot be accessed by anybody. And if anything is ever shared, it's always de-identified. And so in contracts with, let's say, Citizen, for example, we can have that de-identified data moved over to Dennis to then populate into the portal. So there are ways to do it. Citizen, Brian, just specific to you, the way Citizen does it is they charge industry money to get the de-identified data because that's just how they do financially. But the foundations are exempt from that. They're not under that kind of policy. It's a separate thing. So they let us have access to the data. I hope that answered.
I just wanted to add, which we are going to be, I was just going to add that which the Citizen data we have access to, we'll be sharing with Dennis so he can incorporate that. And we've also made efforts to connect Dennis with Ampidori, who ran the study at Boston Children, who he is connected with because I believe he trained with her. So yeah, we are definitely trying to make an active approach in bringing all our data into one place. I also just wanted to call out that Alia had a comment right in the beginning of the chat about that similar tools exist with KCNQ2 from Spain. And so just sort of, maybe international collaboration. I don't know, Dennis, if you can speak to that at all.
Sure. I mean, so any form of data which is de-identified and ideally has a CRID, and where combining the data is straightforward, can be done. And I cannot speak to which degree how the data looks like, but typically that most people, it's not, for most surveys or most data sets, it's not very sophisticated. One would, I don't know if there's a language, yes, there's probably some work for translation. But so as long as everyone is willing to share, there's typically some degree of work to be done, but then it can be combined. So there's ways where people have figured that out in general. And also just to, I'm just looking again at Brian's list from collaborators. So Boston Children's is Anne Poduri, and Anne already contributed to other portals, so such as Cogna1A portal, which we built. And that should not be an issue. Citizen is almost here already. And And Combined Brain sent me this morning their data from another disorder for a different portal. So it's just it's just a matter of months until everything is combined. So I'm very optimistic in this. But of course, I'm just not discussing too much about all the work which is needed to be done, because it takes some energy to bring it together. But these days, everyone is very collaborative, and yeah, I think one can be optimistic.
All right. Well, we're kind of at our time limit, but I wanted to first thank Dennis and his team for joining and being a part of this. And, he's going to do some presentations at the summit, which we are setting up streaming for that. So people will be able to do a virtual registration, and we will also be recording, and then we will be doing updates as data kind of rolls out to the community on where things are, and we'll have those frequently. So there won't be, our big thing is we want to have a turnaround. We know what a commitment it is. As a parent, I understand the time it takes to get all these, to sit down and do an interview and answer a questionnaire. So we're really, really focused on trying to get that turnaround to show families how involved we are in making sure this information is out there publicly and not behind any kind of paywall or anything, so that, researchers can kind of, access, and we build out that, next steps of where we need to go as a community. So, I want to thank everybody all for your time. We're super excited to have you here, and hopefully we'll see some of you at the summit. All right. Thank you so much.
Thanks, everyone.